Hi, my name is Diane Srivastava, and I'm a professor at the University of British Columbia. I study ecology, especially how species interact with each other in food webs. As a scientist, I want to do experiments to test my ideas. But how do you replicate and manipulate an entire food web? Food webs have animals in them that like to disperse out of any sort of small experimental arena you might put them in. So my solution is to use a system that is naturally contained. Introducing the tropical bromeliad. Many species of bromeliad can trap water between their leaves, as well as dead leaves from the surrounding trees that can fall into the plant and start to decay. That forms the base of the food web. The dead leaves are colonized by microbes and aquatic invertebrates. So things like insects, but also worms and ostracods. And these animals become prey for other animals. And so we have a miniature food web that is perfect for doing replicated experiments. To study bromeliads in their natural environment, we work at Patia Biological Station in the Área de Conservación Guanacaste in northwest Costa Rica. Here, it rains a lot during the winter, almost 3 meters per year, filling the bromeliads with water. The surrounding rainforest is rich in biodiversity. It is estimated that 4% of the world's named species exist within 50 kilometers of the station. I've been working here since 1997, and over that time, we've catalogued roughly 100 different insects and other invertebrates living in bromeliads. Our field crew changes every season, but usually consists of local fieldwork contractors like Suhuedi Romero on the far left, volunteer field assistants like Pablo Sandoval in the middle, and graduate students like Nadia Piaz next to me. To set up an experiment with bromeliads, our first task is to find the right size plants. This involves hiking through the rainforest with buckets to carry the bromeliads in, and a collapsible ladder with which to climb trees. The terrain is hilly and muddy with lots of streams to cross. This is definitely rubber boots ecology. When we find the right size bromeliads, the next step is getting it down, which is made easier with our field ladder. The bromeliad just uses its roots as a holdfast on branches, so we can usually pry it off the branch with a knife. The trick is holding it perfectly level while getting it down to the bucket, so as not to lose any of the water or the aquatic insects. Another field hazard are snakes and scorpions that sometimes hang out on bromeliad leaves, like the one silhouetted here. This area of Costa Rica has a high density of venomous snakes, like this Fer-de-Lance that was found under the station and captured in a laundry basket. Once we have the bromeliads back at the station, the next step is to wash out all the water, leaf litter, and invertebrates inside each plant, and then put these through several sieves. We also measure the water holding capacity of each plant. Then we spend hours sorting through the sieved contents of the plant, looking for the tiny aquatic invertebrates. We look for the movement of animals against the white tray and wash off larger pieces of leaf litter that they may be clinging to. When we find an animal, we identify it with a hand lens and put it in a container according to its identity and size. To keep our insects alive, we need to know what it eats and to be sure it has enough to eat. These marsh beetle larvae like to scrape the surface of dead leaves. These mosquito larvae filter feed small particles. Mosquito identification is very tricky and usually requires a microscope. In this dish, we have mosquitoes of several different genera. Tipulate or cranefly larvae shred leaf litter with large chopping mandibles. Although we've also discovered that they can opportunistically consume other insects if the bromeliad dries out. One of the most important predators at this site are damselfly larvae that engulf their prey whole. As adults, these are magnificent helicopter damselflies whose asynchronous wing beat gives them extraordinary agility. Other predators pierce their prey and liquefy and suck out their insides, like this ceratopogonid midge who is taking on a mosquito larvae. We hang the bromeliads upside down to dry to ensure there are no further insects inside them. and then we take them to the field, where we hang them in a string basket. Then we reassemble the food web exactly as we need it for our experiment, starting with a pre-weighed amount of dried leaves, then adding water and invertebrates. When the food web is assembled, we sometimes need to make sure that no other species colonize it, so we cover it with a net. And that's it, one bromeliad experiment ready to go.